day, grade 12 learners. Welcome to today's Life Sciences lesson. My name is Griselda van Wyk, and this lesson is brought to you by the Gauteng Department of Education in collaboration with Sibona Discovery Centre and broadcasted by Bakama Research and Development. If you have any questions, please use our WhatsApp line, and the number for the WhatsApp line is 063 629 3655. I'll just repeat that, 063 629 3655. Okay, so let's get started. Today we're going to look at pedigree diagrams and mutations. So I'll start off with the pedigree diagrams. And as soon as I can get this thing to move, whoops, all right. For every matric learner out there, it is extremely important that you have an exam guideline. I hope by now you have your exam guideline in your book, right? Because this is like a little Bible that you use for studying life sciences. And you will see in the part, and remember it must be the correct one, just check, it must be 2021, not 2017. And this has been done by the examiners for you to make it easier for you to know what exactly it is that you should know about a topic. Now you will see this topic that I'm doing now, genetic lineages and pedigrees or pedigrees. What are they saying? They are saying firstly, a genetic lineage or pedigree traces the inheritance of a characteristics over many generations. We'll get into that just now. And you'll also have to be able to interpret a pedigree diagram. And that's what we're going to be doing this afternoon. Right, now let's talk about a family tree. Now you should have heard about a family tree by now, right? Your great aunt and this uncle and that one was rich and that one was poor and all these things. Now, if you look at this little tree thingy we have here, you'll see that there's a little family tree and the little girl there with the red hair. She's me, but it's not her name. It's just what she calls herself. She's got a little brother and a sister. There's her mother, her father, her grandfather, and her grandmother, right? But she also, her mother has a brother, and that's her uncle, right? And he's married, and they have cousins, right? Or children, and that's her cousins. Now, in sciences, we're not going to draw a little tree every time we want to ask you about a family tree. No. We're going to use a more scientific method. And this more scientific method is using lines. You can see here, we've got lines between grandpa and granny. And this side, because you obviously have more than one set of grandparents. This will be your mother's father and mother, and your father's father and mother, right? And obviously that will be your mother and your father, and this will be you and your siblings, right? So a pedigree diagram or a family tree will mostly look like this. And then secondly, we always look at characteristics like features in a family tree. So when you look at a family tree, you're not looking at things like who's rich, who's poor, or this uncle is very weird. You know, what's wrong with this uncle? Now we're trying to check out in the family tree what's wrong with this uncle or aunt. No, we are actually looking at a genetic feature or a genetic characteristic or an illness in this pedigree diagrams, right, for life sciences. Other people and other professions or so on will look at other things, but in life sciences we are looking at genetic diseases or characteristics, right? So if you look at, this looks very complicated, doesn't it? This specific genetic lineage or pedigree, we can see the difference between a genotype and a phenotype. Now this one here is talking about brown eyes and blue eyes, right? So the phenotypes, remember phenotype is the physical trait, the thing that you can see, okay? So this will be a blue-eyed person and that will be a brown-eyed person, and they have added this to the whole tree, right? The genotype is now the alleles that we are assigning to each characteristic. You can be dominant, and in this case the brown eyes are dominant. And this is very important to understand with pedigree diagrams, is that when you have dominance, there can be either homozygous or heterozygous, and you need to figure this out usually, right? So the dominant homozygous will have two big B's, right? Two capital letters. And the heterozygous one will have a dominant allele and a recessive allele. Can you see that? The recessive trait, in this case blue eyes, will have two. It will always be homozygous, because if you have a recessive trait, you will then have the physical appearance of that recessive trait. And the genotype will then be two small letters, right? And that's the difference between a genotype and a phenotype in a pedigree diagram. But that's not all you need to know. You must also know what does the circles and the blocks and the lines mean, right? That's very important. Now, firstly, when you see a circle, that will always be a female, always. The blocks will always be males. And then if you're talking about a genetic illness or a disorder, it will be the shaded blocks and circles will be the affected individuals, right? They will have the disorder or the feature that you are talking about in this pedigree diagram. Also, a pedigree diagram is not like a Christmas tree. There's not like 50 characteristics that you are looking at in one pedigree diagram. You can't look at eye color, hair color, nose, your nose, the size of your nose, or how tall you are, all in one pedigree diagram. No, we are only looking at one feature or one illness in a pedigree diagram, right? Or else it's going to be very, very chaotic, okay? So, also, when two people had children, we have a line here between them. Can you see between the circle and the block? That means that they don't have to be married, but we will call it married, right? So they linked up, and then this will be the line of descent. And this line, horizontal line here, will be for the offspring, for the children, all right? And this pair have two children. They have a son and a daughter, right? Remember, a block is male and a circle is a female, right? That will be the mother and that will be the father. Another thing that you should know is that the generations are there. This is your first generation, right? And your second generation. If you can go back to our little family tree, you can see this will be our first generation, the grandparents, right? This will be our second generation, and this will be our third generation, right? So you have generations in your pedigree diagrams. All right. So those are basic things that you need to know in order to interpret a pedigree diagram. But let's see now, how would you go about to solve a pedigree diagram? Right. Let's look at this example, for example. We have features, earlobes. Now, unattached earlobes are dominant, right? Now, if you look at my earlobes here, it's attached. You can see it's there. It's fixed to my face, right? So I have the recessive trait. It is attached earlobes. If you have unattached, it's not attached here, it will be the dominant trait, and it will be a big F, right? So what is the first thing you do? Oops, wrong side. You study the key, right? There's the key. Please, people, always look at the key. That is so important. They don't give it there just to be funny, or maybe they didn't know what to do with their time, or there's a blank space open on the page. Now you just put in a key. No, there's a reason why they put it in there. You need to look at that key. So the first thing that you look at is you'll see that the shaded ones have the attached earlobes. Now attached, we know now is recessive. So remember that. And then the unshaded ones will be the unattached. That will be the dominant one. All right. I keep on swiping the wrong way. All right. Now we look for dominant and recessive characteristics, like I said. So with attached, you will be recessive. So attached will be recessive, and unattached will be dominant. All right. It's nice just to write that in for yourself to remember. And then you fill in the genotypes of the recessive ones first, because recessive will always be the easiest. Why? Because they can only be homozygous, right? So in this case, the attached ones is shaded, so we will fill in small f, small f, small f, small f, small f, small f, and this one is also small f and small f. So all the recessive ones have been filled in now, right? They are attached, they are recessive, homozygous, easy peasy, no problem there. Aye. But now we have to see what the dominant traits, or the people with the dominant characteristics, are they heterozygous or are they homozygous? That's the decision you have to make by looking at the recessive ones. The recessive ones will give you the clue, right? So because each recessive person will get a recessive allele from both their parents. They will get one from the mother and they will get one from the father. All right. So you work backwards and you fill in one recessive allele for each parent. So let's start here at the bottom. All right. So this little girl here, remember it's a girl, she received an allele from the mother and an allele from the father. Now this girl has the dominant trait, right? Because she is unshaded. So it's unattached here. So it's dominant trait. So she obviously got the dominant allele from her mother, which is F there. And from the father can only give you a recessive in this case. Look, he does only have 2 recessive alleles. Where will he get a dominant allele now? It's not going to fall out of the sky. No, the father gave her the recessive allele. So that one is heterozygous. All right, let's move up one here. And we look here at this individual here. Right, again, he can only get a recessive allele from his father. He can't get a dominant one there. And he's got the dominant trait, so it's heterozygous again for him, right? And then this mother here, if you look at the mother has a child that is recessive. Can you see there? Right. Which means this child had to get 1 recessive allele from the mother and 1 recessive allele from the father. So it means that this mother must have the recessive allele. Right, but she's dominant, she's got the dominant trait, so it will be heterozygous again. All right, let's look at the last one here, the mother here. Again, this mother has a child that is totally recessive. So she must have the recessive allele to give it to that child there. So this mother is also heterozygous. Okay, so that's what we mean by working backwards. You look at the children, you look at, is it a recessive child? Because that means that if it's a dominant parent, it will be heterozygous. All right, let's move on. I've already said that, and I've already said that. Right, so that's a short way of looking at it. Let's look at the next example, and that is lips. Now, this pedigree is all about broad and thin lips. So if you have luscious, big, broad lips, you are dominant. Right, dominant trait. So this guy here, male with broad lips, will have the dominant trait. Thin lips like mine, again, recessive. I'm like a recessive person. Then they will have a small b. All right, small b. So, and I just want to reiterate this. It is so important to read the STEM part of the question. If you go to your question paper and you see a pedigree diagram, some children are like so lazy. They just look at this tree and maybe, if we're lucky, the key, and then they go on. They don't read what is stated above that thing. And there's so much information in this sentence here because they are telling you who is dominant. They are telling you who is thin lips, which one is recessive. And they're also telling you something different here. They're saying individual 2 is homozygous. Right, homozygous. Now, individual 2 is shaded. And if you look at the key is saying that if you shade it, you have broad lips. And broad lips is dominant. So this one will be then big B, because that's homozygous. That's what homozygous means. Right. But now you have to go and work out the phenotypes and the genotypes. Right, now the phenotypes are very easy because the unshaded ones will be thin and that one will be broad, right? Broad, it's so easy. So if they ask you something like this, you just know, just look at the key. That's so easy. Phenotype, what you're seeing. Thin, okay, I'm just going to write broad, thin, 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 and broad. Okay, easy. That's not difficult. You just have to interpret the key. Very easy. They won't give it to you like this in exams. They won't expect you to do all the genotypes and all the phenotypes. But I'm just doing it this afternoon just to show you, the different possibilities and so on. All right, now we need to get to the genotypes, and that's the difficult part. Now we can work our way back. So, if a child is recessive, he's got the thin lips, it's very easy. It's small b, small b. So you do the recessive ones first, always, right? Because that's easy. They can only be recessive, right? So all the unshaded ones are small b, they're recessive. It's the shaded ones that are now tricky because you don't know, are they hetero or homozygous? All right, so that's where you start working back. So now we look at this boy here. This boy has a father that has the dominant trait, and it has a mother that has the recessive trait. So the mother can only give him a small b. Only can give him there. So now we already know that this one will be heterozygous because he's got the dominant trait. Same here. Exactly the same with the sister here. Right, because it's getting the recessive one from the mother. All right, from the mother. I think I've done, okay, yeah. The broad ones here, both of them, again, the father has the recessive trait in this case, so the father can only give them a recessive allele, which means that they are heterozygous. Right, and I think I've done all of them. Can you see how easy it is to figure out the genotype of an individual in a pedigree diagram? You always start with the recessive ones first, and then you work your way backwards. Whoops, wrong way. All right, now we get to color blindness, which is a sex-linked allele. Right, now we have problems with this one every year. Children think when they see a pedigree diagram, it's sex-linked. Always. No, it's not. You have to see what the question is telling you. If it's colorblind or hemophilia, it is sex-linked. Or if they tell you in the question, this disorder is sex-linked, then it's sex-linked. But now with the broad lips and the earlobes, it had nothing to do with sex links. Did you see? So please, if you see a pedigree diagram, don't immediately think, okay, now I'm going to do X's and Y's, because that's what you do. We see it every year. Right, so let's look at the stem of the question, or what they're asking. The following pedigree diagram is for color blindness. Determine the possible genotypes. Genotypes for numbers. So I don't want the phenotypes, I want the genotypes. Right, they tell you here, and that's very nice of them. They don't actually have to tell you that it's sex-linked, because you're supposed to know that color blindness and hemophilia is sex-linked. But they actually tell you that. Oh, they were feeling very generous that year. Right, and they also tell you this, that the affected allele has the small b. Can you see? It's affected, and the unaffected one, or the normal allele, is the big B. And just a reminder that males will always have an X chromosome and a Y chromosome, and females will always have 2 X chromosomes. Don't confuse it. All right, please. That is how males are biologically or genetically. They've got an XY chromosome, and females have got an XX chromosome. All right, so that's how this is going to work. All right. So you have to have now to look, if an affected male, look at the key, color. It's shaded. It's shaded, right? It will be shaded, so that person will have the affected alleles, right? Normal male will not have it. Remember, what is sex-linked? I'll just refer back to it. The allele is carried on the X chromosome only, or the gene is carried on the X chromosome only. It's not carried on the Y chromosome. So when you have a situation where this chromosome has the affected allele, like a small b, then the Y chromosome does not have another allele to mask it. So that male will definitely have color blindness. Definitely. Right. There's no way that he can have a dominant allele because he doesn't have it on his Y chromosome. Whereas with females, you have a chance, if you have a small one, to get a dominant allele, or you can have a recessive allele. So your chance of getting the sex-linked illness is much smaller than the males, because you've got 2 X chromosomes and both have an allele for the illness. All right, so just keep that in mind. So just let's look at where we will start here. Usually with sex-linked diseases, you start with the males, because the males are so easy, right? Because they either have it or they don't. So now you look here and you see, right, click harder. Oops. Okay, there it is. The normal or the unaffected males, the males without, you must actually say, don't say normal, you say unaffected, because the key said unaffected. Okay, so he's unaffected. He will have a big B. Can you see? He's not affected. Right. So the same will be happening with the other male there, number 11. What am I doing? Oops, sorry. Just make it small again. And just swipe to the left. Can you see that he also has the unaffected allele? Because he's normal, he's unaffected. He's not shaded. Now the shaded males will be the small b. So you can fill in all the males, yeah, well, that's affected there. There are, it's easy. The problem comes in with the women, right? Because now the women can either have they can either be heterozygous or homozygous, and you have to figure that out. Now, I'm going to tell you today it's very easy. When you look at a pedigree diagram, look at the mother, right? Does she have an affected son? If she has an affected son, it means that she is definitely a carrier or she's heterozygous. She's got that recessive allele. Now, let's look at number 1. Can you see number 1 there? She's got an affected son. Number 5 is affected by this. So where did he get that affected allele? He couldn't get it from his father. The father is giving him the Y chromosome. He can't get the Y chromosome from his mother. It doesn't work like that, hey? You must know that by now. So the father can only give the Y chromosome. So where did he get that affected allele? From the mother. Right, so the mother donated that affected allele. So that is why the mother will then be heterozygous. She will have the dominant as well as the recessive allele. Number 3 there, same case, look at there, she's got an affected son. So, keep on swiping right. So can you see that she's also affected? She's also got that, not affected, she doesn't have it because in her case, she's got a dominant allele that is now masking the recessive allele, right? Let's look at number 6. If you look at number 6, the mother of that son there, same thing. Me and this swiping. The same thing, because she's got an affected son. Okay, this lady here, number 8. We don't know what number 8's children look like. But now let's look at her parents, work backwards. We see that she's got a mother who's got a recessive and a dominant allele, but the father is affected. What can the father only give to this girl? An X chromosome. He cannot give the girl a Y chromosome. Why? Because it's a girl. She's not a boy. So the affected allele will come from the father, that one there, but she's not affected. So she got the dominant allele from her mother. All right. Last one here, number 10. If you look at number 10, you look at the parents there, again, here's the same situation. The father has a affected allele, but the mother saved her in this case, and she's heterozygous. She's not affected. So this type of family here is the type of family you do not ask to come and rearrange your sitting room and do the color matching of the couch and the curtains. There's a lot of color blindness going on here. So rather, maybe the girls, but not the boys at all. Right. Right, but let's have a look at how they would actually ask it in an exam situation. So we have got a question from paper 2, November 2020. And let's see what they ask. Gold syndrome is a sex-linked. That's why I say always read the stem of the question, the top part, yeah. Don't skip it and just go there because you won't know what's going on. They are telling you this one is sex-linked. So you have to use X and Y, yeah. Right. It is caused by a dominant allele. Look at this one. This is a dominant one. Color blindness and hemophilia are both recessive, but this one is dominant. So even if the lady gets it, she's got it. The diagram below shows the inheritance of Gold syndrome in a family. So I don't know what Gold syndrome does to you, but this family is quite affected by that. Right. Name the type of diagram shown. Is this a difficult question, people? No. Hopefully you will immediately know this question, that this is a pedigree diagram. So easy. Yeah. Now I want you to count how many females are in this family. Right, I want you quickly to count. I'm going to give you 10 seconds. Quickly count the amount of females in that family. 5, 4, 3, 2, 1. All right, let's see. What did you get? Well, let me tell you, they really, really got me on this question because I was counting the circles. I was saying, right, 1, 2, 3, 4. And I thought, yeah, I'm right, 4. Nope, they were catching you out because actually, if you look down here, they have 2 females here as well. So that also teaches you a lesson. Be careful, read carefully, look in detail at the pedigree diagram. Don't just skip now quickly. I know circles are females. They wanted to catch you out here. All right. So it will be 6. 6 females. All right. How many males in the F1 generation have Gold syndrome? Right. Now they are seeing, do you know how to link F1 with a pedigree diagram? So now you know what an F1 is. It's in your genetic crosses, and I hope you've been doing a lot of genetic crosses these past 3 weeks. I really do hope that. That is your first line of offspring, or your filial 1 generation. That's the first, the children, the first line of generation. So your P1 will be the parents. So P1 will be there, and this will be your F1. So firstly, they're testing, do you know that? Okay. Secondly, now they want to know, can you read the key? Because the key is telling me that the affected males will be shaded. How many are affected here of the males? Well, there are only 2 males, but only 1 is shaded. So, and that's Pelusa. So there will only be 1. Okay, let's go to the next part of the question. Give Gabby's genotype. Now let's look at Gabby. Gabby is affected. Right. So we already know that she, okay, let me just quickly look. The key here is X, G. Yeah, but now we don't know, is she hetero or homozygous? But she's got the dominant allele, so it must be a big G. So now let's have a look at her children. That's the easiest way of looking at it, right? So if she's got a girl that is unaffected, unaffected means that this girl had to have 2 recessive alleles. Right, where would she get these recessive alleles, right? She has to get 1 from Gabby, and she has to get 1 from her dad, which has the recessive trait. So in this case, she will then have a small g there. She will be heterozygous, all right? Keep on forgetting. Okay. Anu and Pelusa have 4 children. Give the phenotypes of their son. I just want you to have a look at the sons. Where's Anu and Pelusa? They are here. There's Anu and Pelusa, and they have 2 sons there. Now you have to go and think, what are the phenotypes? What is phenotype? Physical. It's the physical appearance, right? So that they were clever here. They didn't give you the key. Now you can't use the key because it's just written in words. Now you have to go and figure out what will be the phenotype of them. Right, so must I go on, or do you want some time to think about it? Maybe I will give you a minute. Take a minute for that one. Okay, I hope you didn't slack off now, start sleeping or ran away or something like that, that you actually tried this. But let's have a look at what happened here. So we want to know what is the phenotypes of their sons. All right, so we want to know what they look like. Remember, this is a dominant illness, dominant. So let's look at the mother. The mother is unaffected, which means she cannot have the dominant allele. She cannot have it because she's unaffected. The dad has got the affected allele, but he's got a Y. So what does the dad give the sons? He gives them a Y. So he couldn't give them the affected allele at all. He's only giving them a Y chromosome. Right. So if they both have the Y chromosome from their dad, what are they going to get from their mother? The recessive allele. Right, so they're going to get the recessive allele. And if he gets the recessive allele from his mother, it means that he is unaffected. All right, so that one, you had to think a bit there. And now they want to know explain your answer. And that's actually what I just said now. So Palooza, the father, is affected. He's got that allele there, right? But they're giving the tick there. Can you see before that? So you can either give that or say it's affected. Anu, or the mother, is unaffected. So she's got both those things, the recessive alleles. Males inherit the Y chromosome from their father, or Palooza. So they inherited the recessive allele from Anu. Right. And people, another thing that I also want to emphasize here, please don't just talk about X. Like males inherited the Y from Palooza. They're not going to give you the mark there. The Y what? You must say Y chromosome, right? Or X chromosome. It's not just an X. I know we get, now you start talking like that, now you're pro and you just say, yeah, the mother gives the X and the father gives the Y. But if you just say that in an exam paper, you lose marks. They don't give you the mark because what is an X and what is a Y? You have to specify X chromosome and Y chromosome. Ish. Me and this scrolling. I'll get it right just now. Right, this is another, I like this activity because it shows you how a recessive condition can come out from parents that does not have their condition. Let's have a look. They say diagram below shows the pattern of inheritance of a disorder, right? Then they say unshaded is unaffected, not affected, but the shaded one is affected. Now look at this. Both parents are unaffected, but their daughter is affected, which means that they have a allele there that is masked. It's not, you can't see it in their phenotypes, in their physical state. So it means that allele must be recessive, right? So they both have the dominant, let's say B, right? I'm just making an example. But they both have a recessive allele there that they gave to their child, and their child is now affected with a recessive condition, right? So when you have dominant parents that have a child with a recessive condition, you know that they are both heterozygous. They have the recessive allele there. So the answer here would be A, right? This one, recessive allele. We know it's a recessive allele. Both parents are heterozygous. I'm not going to go through the rest to save some time here. If I can just learn to swipe the right way on this tablet, it will go faster. Right, now we've got a next pedigree diagram, and they talk about a dominant allele causes the last joint of a little finger to bend inwards towards the fourth finger, and they call it B, big B, right? And it's bent little finger. So some of you are walking around with bent little fingers. The recessive allele, small b, cause a little finger to be straight. Now I'm again recessive. Look at my fingers here. I don't have a bent finger. Can you see? It's straight, very straight there. But a bent one would have been like something like this. I'm not going to break my finger for you, but you can. I think you've got, some of you have it, right? So the bent one is a dominant one, and the straight one is a recessive one. All right. Now they say, the pedigree diagram below shows the inheritance of a bent little finger family. So this family, some of them have bent fingers and some don't. Now, the straight finger, can you see the straight fingers are the recessive ones, and they are not shaded. So you know all these ones here that are unshaded are straight fingers, right? Then the bent fingers were the dominant ones, the big B's, they are all shaded. But they don't want all of their phenotypes or genotypes, they just want certain ones. So they say, explain why individual A and B are definitely heterozygous for this trait. So let's have a look at A and B. Now, if you look at A and B, that is the grandparents. They're a grandmother and grandfather. Why are they now heterozygous? So they're telling you that they have both alleles, right? Why? Look at the children. They have both. They have 2 children with the recessive trait. Just what I said now. So where did that recessive alleles come from? The child had to get 1 recessive allele from the grandfather and 1 recessive allele from the grandmother. He is a small b, and this one is also small b, small b. So that small b had to come from one parent and the other one from the other parent. So that's why you know it's heterozygous. But let's look at the nice explanation. The parents have the dominant phenotype. Both of them are dominant, right? So they got the bent fingers. They have children with a recessive characteristic, and the other allele from the parent must be recessive, right? So they have the dominant allele, but they have a child with the recessive alleles, so their other allele had to be recessive. Oh, man. All right, now this next question, they're asking you, individual C has a child with a partner that has straight little finger. What's the straight finger? Recessive. Remember? Use a genetic cross, well, that's the thing, yeah, but we'll just get into it now, to show the possible geno and phenotypes of the child. Now, I'm just going to go back to the question here. Where's C? Yeah, is C. C has the dominant trait. So C has a big B, but we don't know, is it hetero or homozygous? So now they want you to figure that out. That's why they put it in a pedigree diagram. So they're making it a bit more difficult. They want you to be able to think. Okay, so how do we do that? We look at the parents, right? So now we look at the parents. If that parent gave him the dominant one, this parent can only give you a recessive allele. It can't give you a dominant allele. It's only recessive. So this will be a heterozygous one, all right? So this is just a normal cross. You'll have your P1 and your F1. This F1 must actually be, yeah. The phenotype, I told you, bent little finger and straight little finger. Remember, he's marrying a straight little finger person, but C's got a bent finger. We just figured out that C, this is C, right? We figured out C is heterozygous, and obviously straight is recessive, so that cannot be anything else. You know meiosis, you know how to get to the gametes, that you have to show that they are gametes. They must be separated by a comma or something to show that they are separate. And then you can choose two ways to do the fertilization bit. You can do the crossing over bit, which I don't like a lot, but it's up to you. I love the punnet because you just put the one set of gametes in here, right, and the other set of gametes there, and you cross them over big B, small b, small b, small b, and this side, big B, small b, small b, small b. And there's my F1 genotype, and that's what they asked for. They asked for the genotype. And in this case, you'll see there's half of them will have bent little fingers, and half of them will have straight little fingers. So that will be your phenotype. You do not have to give this ratio of the offspring here because they didn't ask for it. But if you want to, you can do that. All right. Next question. One type of deafness in humans is carried on a single allele. The diagram below shows the inheritance of deafness in the family. Now, I'm going to give you some time to do this, but I want to just to say the following. Is this sex-linked? Are you going to do X's and Y's here? Think about it. Just think about it. All right. Let's give you a minute. You don't need more than a minute for this. So did you think about what I was asking? I was asking about, is this sex-linked? Are you going to do a lot of X and Y's here? Now let's look at the question again. One type of deafness in humans is carried on a single allele. The diagram below shows the inheritance of deafness in a family. Is there anything about sex-linked here? No. So you do not do X and Y's here at all. Okay, not at all. But let's look at the questions. How many generations are represented in this pedigree diagram? Remember what I said in the first slide. You look at how many steps there are or how many levels there are. So this will be the grandparents, this will be the parents and the aunts, and these will be the children. So that will be three generations. All right. The children of Paul and Lizzie are able to hear. Or how many children of Paul and Lizzie is able to hear? All right, now there's Paul, there's Lizzie. How many of their children will be able to hear? Now that's the easy one. What must you do? Go and look at the key. It's easy as that. Easy peasy. Look at the key. Unshaded is hearing. Shaded is deaf. So how many of their children will be able to hear? They've got 1, 2 unshaded children. Right, so it would be 2. Right? Which phenotype is dominant? All right, so now we have to go and look here at the different, you have to work backwards. Now, if you look at Paul and Lizzie, they are both have the different phenotypes. So their children won't tell you anything because they've got, you know, the one has got the recessive and the one has got the dominant. The same with Mary and John. But Bob and Anne, they have exactly the same phenotype. So if they have a child with a different phenotype, you know that that different phenotype must be the recessive one because they are not showing it in their phenotype, but they are masking it in their genotype. Right. So if we look at Bob and Anne's children, aha, there they have a deaf boy, right? A boy that has deafness. So that child had to get 1 allele from Bob and 1 allele from Anne. They are not affected, but they have an affected child, which tells you that the dominant phenotype here is hearing, right? But that the recessive one is deafness. Okay. So hearing is the dominant one. Use the offspring of Bob and Anne to explain your answer to question 4.2. Whatever, the previous one. That's exactly what I actually said. Bob and Anne can both hear. There they are. Both of them can hear. They have a child who is deaf, right? And this means that each parent carries an allele for deafness, right? But it's masked in their own phenotype. They're not deaf, but they are carrying an allele for that, which means they are heterozygous, and that hearing is the dominant characteristic here. Use the letter A to represent the dominant allele and small a for the recessive allele to give all the possible genotypes for hearing individuals. Now, people, when you have a dominant characteristic, there's only 2 ways, and I said that on slide 2. You'll either be heterozygous or homozygous. So easy as that. That's what they are asking you. So what you're going to do is you're going to just give them the homozygous, big A, or the heterozygous, big A, small a. It's easy as that. That's what they are asking here. Right. Hemophilia. This sounds familiar. This is the one that they are talking about when they're doing sex link. What did I say? You have to know this one. This is it's prescribed in the exam guideline. You have to know this one. Right. So you must know it's sex linked. You must know it's recessive. Right? They're not going to tell you in the question. You must actually know it. But I think in this question they were actually feeling sorry for the children and they gave it away. The following pedigree diagram shows the inheritance of hemophilia in the family. Remember, that's that bleeding disorder where the blood doesn't clot and people can die, bleeding out and so on. Very painful as well and so on. Determine the possible genotypes. Read the question, underline what they want for letters A, B, and C. Use, that is our X small h for the affected allele. It's also telling you there that it's recessive. And X big H for the normal allele. Here they are using the word normal. Always use the words that they are using in the question. If they said affected, unaffected, then you use affected, unaffected. If they use normal or not normal or whatever, then you use the words they are using. All right. So now let's look at number A. They want the genotypes for A, B, and C. If you look at A, right, that's the mother. Right. Now remember, women can be carriers. They can be heterozygous. She's obviously not hemophilic if you look at the key. She's not affected. Right. She doesn't have hemophilia. But now we must look at her children. Does she have an affected son? Now let's have a look. Yep, there he is, number B. Number B is affected, right? So if he's affected, where did he get that recessive allele from? He got it from his mother, because he can't get an X from his father, X chromosome from his father. He can only get a Y chromosome from his father. Even though his father is now affected, his father's got hemophilia, he did not get hemophilia from his father. He got it from his mother. Shame, imagine now in this case, everyone is blaming the father, but well, we can't assign blame, but I mean, everybody's thinking it's the dad, but actually the affected allele comes from the mother, right? So this mother will have, she's not affected. She'll have the dominant allele, but she will also have the recessive allele. That will be number A, right? Because she's got an affected son. What does the affected son look like? Right? He will have the recessive allele and a Y chromosome, not an X. If it's two X's, it's a girl. It's no longer a boy. You can't go there. Don't change the sex of the poor person. Number C, the daughter here at the bottom. Oh, you have to go and look now. Work backwards. What about the father and the mother in this case? Right, this child is not affected. So this child must have the dominant allele, because you cannot see that there's hemophilia. There's no hemophilia there. But just have a look. What did she get from her dad? The dad is affected. What is the dad giving the daughter? Not his Y chromosome. He's giving his X chromosome. And his X chromosome is affected. So he will give a affected allele there, or the recessive allele there. So she will definitely be heterozygous. Right. So remember with color blindness, hemophilia, any sex-linked, if you're not sure about the mother, look at the son. Look if the son is affected, right? And if you have a daughter and you don't know, there's no children, she doesn't have children yet, then you look at the parents. See which one could give her the affected X chromosome, right? Not the Y, has got nothing to do, it's normal, right? So I think that's the end of my presentation. We will be taking a 5-minute break now, and then we will continue with genetic mutations. So 5 minutes, don't run away and don't come back. You come back, you go and drink some cool drink, water or whatever, eat a piece of bread or something, but you come back. We will see you in 5 minutes time. Goodbye.
Good afternoon, matriculants. You are speaking to Sina Sekheche. Welcome to the second session, which is mutations. But before we continue with the session, please remember our WhatsApp line. You can ask your questions on WhatsApp. The number is 063-629-3655. I repeat the number. It's 063-629-3655. Right. Now, starting with our second session, which is mutation, we're going to go through our exam guideline to check the scope of this topic. So before you start any topic, you need to revisit your exam guideline. The exam guideline is going to tell you the scope or how much you should know of the topic you will be studying. So when we take a look at this section, you have to know the definition of mutation. You need to define mutation in general because there's other types of mutation that you need to know the definitions for, and you need to know the effect of mutation thereof. There are harmful mutations, harmless mutations, and useful mutations. Then you come here, you define how mutation contributes to genetic variation. I know most of you are familiar with this one, as you did meiosis. You know how genetic variation comes about. So we're going to see how mutations also brings about genetic variation. You also need to know now, this one is very, very important. Please highlight this one on your exam guideline. You have two types of mutations, which is gene mutation and chromosomal mutation, which are different from the first one, the general term of mutation. So you need to take note of these ones, but don't worry, we're going to explain them to you further. Then you also need to know the two types of mutation that can alter the characteristics leading to genetic disorders. Some of these disorders, you have covered them while you were doing your pedigree diagrams or genetic lineages, but now we're going to look into them as to how mutations cause these diseases. Now, the first one is caused by gene mutations. Now, these are the two examples that you have to know when you get into your exam, which is hemophilia and color blindness. These two diseases, you have covered them as sex-linked diseases in your pedigree just now. But now we're going to look at them as to how are they caused by mutation. Why do we say that they are caused by mutation or gene mutation for that matter? Then you're going to look at another type of mutation, which is chromosomal mutation. And as an example of chromosomal mutation, you need to know Down syndrome. So without wasting any time, let's go straight to our definitions. So, but before you get into the definitions, let us look at some exam tips that you need to take with you when you enter the exam. As some of these exam tips comes from the mistakes that were done by the previous candidates, those who came before you. So we need to rectify those mistakes with you. So the first one is, I have already explained that you need to know the differences between the terms mutation, gene mutation, and chromosome mutation. Most of the learners make a mistake when it comes to this one. They think when you speak about mutation, it's gene mutation all around. So you need to specify. You need to check the question that you are asked, whether they're asking for gene mutation or whether they're asking for chromosomal mutation or whether they're asking for mutation as a general term. So you're going to have to know those three definitions by now. So the definition of a mutation is more general. This is what I am referring to. It is more general than the definition of gene or chromosome mutation, which are more specific as to where they happen. Then please note that when you get to your notes, when you get the resources that you use, they will have further explanation on frameshift mutation or point mutation. Note that this is not required by the new exam guidelines. So you don't need to look into detail of point mutation and frameshift mutation. So don't look into that. So let us look at the terms now in depth. We're going to start with the first one. Your first term is mutation as a general term. Mutation as a general term refers to a sudden change in the genetic composition of an organism. So it is a sudden change in the genetic composition of an organism. So the composition is not specified here, whether it is genetically or it is chromosomal. Then we come into gene mutation. Your gene mutation is where now you need to specify where the mutation takes place. This was a question in last year's paper, and most of the learners got it wrong as they only explained mutation in general. They lost so many marks because you need to specify this word now. Let us take a look at the term in depth. Gene mutation can be described as a change in the sequence. Now, this is very important. The sequence, when you were doing DNA in the first term, you know how the nitrogenous bases are sequenced and how they pair up. So with mutation, you have to state that it is a change in the sequence of the nitrogenous bases or the nucleotides in DNA. If your answer does not have all of these key factors, it is not describing gene mutation. You may find yourself that you are describing mutation in general. So please make sure that when they ask you about gene mutation, you speak about the change in the sequence of the nitrogenous bases or the nucleotides in DNA. Then you will be speaking of gene mutation. The second one, the type of mutation that we need to look at is chromosomal mutation. This one can be described as a change in a normal structure or number of chromosomes. So we're going to look into detail as to how this one happened and the examples that fall under chromosomal mutations. But just to recap, you have mutation, which is a general term. It is a change in the genetic composition of an organism. Then you have your gene mutation, which is a change in the sequence of the nitrogenous bases or the nucleotides of DNA. Then you will have your chromosomal mutation, which now speaks to the change in the normal structure or the number of chromosomes. When we look at the functions, then you're going to know exactly what we look. When we look at the examples, actually, you're going to see exactly which one is which. You'll be able to identify. Thereof, you'll be able to define them. But as we look back, or as we looked at the exam guideline, it required you to know the effects of mutations. Now, these mutations, not all of them are harmful. You may find that some mutations may have actually a survival advantage for some of the organisms. So this can also be seen in bacteria, in viruses. Those, they benefit more on mutations as it happens more with the smaller organisms. Then if there is no selective pressure from the environment, you will find that that mutation will remain in the population. So then you get to your harmful mutations. Some mutations may be harmful. Cystic fibrosis and sickle cell anemia. The cystic fibrosis, these are diseases now. Some mutations may be harmful in such a way that they cause diseases. So one disease that may be caused from a harmful mutation is cystic fibrosis, where you get fibroids in your organs. Mostly you'd find females in their ovaries and their uterus, they will have the fibroids. And sickle cell anemia. Sickle cell anemia is the disease where you find the red blood cells are in a sickle cell shape. And then the disadvantage thereof would be the person would have restricted blood flow or oxygen, because you know the red blood cells, they transport blood, oxygenated blood. So your organs may suffer if your cells are sickle shaped, like a sickle, you know, that C. So that disease comes from mutation, and it can be very harmful. Then some may also form dysfunctional proteins. Then albinism also comes from mutation. And it can be a harmful effect due to the fact that melanin, we know melanin is very important for us because it helps to protect us from the sun, from the UV rays of the sun. So if you don't have melanin, you are more exposed to the UV rays of the sun, and it may cause skin cancer or other types of cancers. So that is a harmful mutation. Right? Mutations, remember we said they are not always harmful. Some may be beneficial. So most of the organisms that benefit from these beneficial mutations are your bacteria. You'll find that some bacteria grow resistance to antibiotics. So it is because they mutate. They form, there's a change, or there's a change in the change in the sequence of the nitrogenous bases that can be beneficial and make them more resistant to antibiotics, and then they grow more, they become better, they become superficial, like your superbugs, MRSA. That is how it came about. It actually was a normal disease, and then it just it mutated and formed a superbug that is not treatable by any means of antibiotics. And then another one is HIV. We know even today it is very difficult to treat HIV due to this mutation also is very beneficial to the RNA viruses because it makes them to not be detectable to the human white blood cells or the antibodies. Then they are not able to detect it and fight it. You know how your antibodies work. When you learned in grade 10, you did the whole diagram on how the antibodies attack the diseases, phagocytosis and whatnot. So with this RNA viruses, you see that it is mutating each time, so it tricks our white blood cells into fighting the diseases. So that is very useful. Hence, we said it is useful to viruses and bacteria. Right? Then there are other mutations that are just harmless. Remember, we spoke about those ones that are harmful and then those ones that are beneficial. Now we're talking about those ones that are just harmless. So these ones maybe they are harmful or they are neither harmful or beneficial to the organism. So they may be important in an evolutionary sense. You may find that they bring about a characteristic that we can study when we study evolution, evolutionary characteristics being inherited, but it does not have any type of effect on the organism or anything. Some are just silent mutations that you cannot even see. They are not observable. They are not. It is virtually impossible to detect those type of mutations. You may find that an organism has mutated, but the way in which this mutation is virtually impossible to detect that there was a mutation. Everything continues as normal. So those types of mutation also occur. Then, looking deeper now into the examples of gene mutation. Remember, we spoke about the two types of mutation. We said it's gene mutation and chromosomal mutation. Now we're going to look at the two examples of gene mutation, which I mentioned earlier that it is hemophilia. Right, we're going to start with hemophilia. Remember the explanation. Gene mutation, it is the change in the sequence of nitrogenous bases or nucleotides in DNA. This definition stays as it is from the exam guideline. Please go through your exam guidelines to check this definition and write it as it is for you to obtain those full marks in the exam. So what is hemophilia? It was already explained in the previous slides that it is the absence of clotting factors. You see those people who have bleeding problems when they bleed or they have something, they cannot stop. When you take a look at this, the first three red blood cells, you see that this is a blood of a normal person, and then the person gets a breakage or a hemorrhage. They break they bump into something or what, they bleed, meaning their blood vessel gets broken and then blood comes out. And then after some time, the blood needs to stop, meaning the coagulation factor is going to come and stop, meaning the clotting is going to take place. The blood is going to stop flowing. This is under normal circumstances with a healthy person. Then when you come to a person with hemophilia, it's a different story. A person will have a normal blood vessel, and then this blood vessel will get broken as they bump into something or they hit themselves, whatever. Then the person will continue to bleed because they have inability to clot. Now, all of this is due to mutation. There is something that happened wrong in their genes. So let's take a look what happened in the people with hemophilia. So with people with hemophilia, you see that there's a change in the F8 gene. Remember, we said hemophilia is an example of gene mutation. So we want to see the problem, the change in the gene. So here they are specific to tell us the F8 gene are the ones that are responsible for hemophilia A, while the mutations of F9 gene or the changes on the nitrogenous basis of F9 gene causes hemophilia B. Now, this is where we find the people. This is how now we find the people getting hemophilia. So the F8 gene provides the instructions for making a protein called coagulation factor. Remember, we said these people, they are anticoagulants. They have anticoagulants. They cannot clot. So F8 gene is the one that carries the instructions that make a protein called a coagulation factor. So if there's a change in the F8 gene, obviously now this person will not be able to clot. So that is due to gene mutation. I think everyone now understands what hemophilia is better, and also why is it an example of gene mutation. It is because there's a change in the gene. Another example of gene mutation that we're going to look at is color blindness. This one is very funny because tests have proven that males and females don't see color the same. But there are those people who have that mutation, that error that happened in their genes when they were sequenced, or were in their nitrogenous bases when they sequenced. You find those people, they have the absence of the protein that make up either a red or a green color. So the cones and the photo, you will be doing the human response to the environment. When you do the eye, you're gonna understand better the photoreceptors that we are talking about here, which are your cones, which you find in the retina. So when you do that, please remember that some people, they may have a problem there. They may not have a protein that make up the red or the green color. So when you take a look at this picture at the bottom here, you see a person with normal vision, the top one. An apple that is green, they're going to see it as green as it is. And then the yellow one is yellow as it is. And then the red one is red as it is. So this is a person with normal vision who has the protein that can make up the red and the green color. But then when you look at the picture at the bottom, this person is colorblind. To them, a green apple is yellow, a yellow apple is yellow, a red apple is still yellow, because they do not have the protein that make up the red or the green. So they only see yellow. When they look at green, it's still yellow. When they look at red, it's still yellow. So it is because they do not have that protein. It is absent, that protein that makes up this color. So this is now why it is a gene mutation, because with proteins, it happens at DNA level. Remember your nitrogenous bases, right? So that is why colorblind. So this is just a picture to show you the picture of the eye, but believe you me, you're going to understand this full picture well when you do the human response to the environment, when you study the eye. But when you look at this part, this is the part that is called your retina, and then it has your rods and cones. So these are the photoreceptors that we were talking about. So that gene mutation will be taking place at these two parts. Then going to a second type of mutation. Please don't confuse the examples of gene mutation as the types of mutations. They are not the types. When we speak about color blindness and hemophilia, they are examples of gene mutation. Now we come into the examples of chromosomal mutation. We said chromosomal mutation can be defined as a change in the normal structure or the number of chromosomes. So now we have to see this. Let's see this in action. When you take a look at this picture here at the bottom, it shows you how a structure can change of a chromosome, or how a number can change of a chromosome. So when you take a look at this little box here in the middle, it shows you the normal set of chromosomes. So, but let's look at how the numbers can change. How the numbers can change for it to be a mutation. If there is an extra number, or if there's a number missing of a chromosome, it is a mutation, meaning there is a change in the number of chromosomes. That is why it is a mutation, because under normal circumstances, you don't expect any change. So if this is a normal set, let's take a look. I have to look at this diagram. If this is a normal set, there are three things that can happen to it in terms of number. First, you may see here you have 2 orange chromosomes, but here you have an extra chromosome there adding. So meaning there's an extra chromosome there adding to the number of chromosomes, which is not normal. Or another thing that could happen is there could be 1 chromosome missing there, which was supposed to be there. It could just move away. Then you know it has mutated. The numbers were not supposed to be like that. This is now the scenario. Another scenario, you can find that you have your initial set, that was set number 1. Then you have an extra set that was not supposed to be there. So that is changing now the number. It is a change now in the number of chromosomes. So that is how now mutation can take place in terms of numbers. Then when we come to the structure, when we come now to the right here, the pictures here in the right, you're going to see that if this is a normal set of chromosomes, something can happen to this last chromosome here. this last part can be missing, like it's cut. So then it is deleted of some sort. So now there's a change there on that number, on the structure. The structure is no longer normal. Hence we say it's a change in the normal structure of an organism. Or this structure can be, the chromosome can actually be elongated or can be amplified. So it was not supposed to be this long here, but it is very long, meaning it has mutated in structure. Then the last thing that can happen, a scenario, is a flip. You can see that here the chromosomes have flipped. If you see on this green chromosome here, there's a pinkish color, then there's a purple one at the bottom. But on the second one, it's a purple one and a pinkish one. So under normal circumstances, that was not supposed to happen. Then it means there's a change in structure. But I think you get the gist of what chromosomal mutation means and how it can happen. It can happen in these different ways, right? Then, as an example of chromosomal mutation, we're going to look at Down syndrome. You did Down syndrome when you were doing meiosis. You know how Down syndrome comes about when you were doing non-disjunction at chromosome number 21, trisomy 21 leading to Down syndrome. So what happens with Down syndrome? Down syndrome occurs due to an extra copy of chromosome number 21 as a result of non-disjunction. So this part we have covered. So when you take a look at chromosome number 21, this is your karyotype of a person with Down syndrome. You're going to see that chromosome 21 has got 3 pairs, and under normal circumstances, this is not right. You need to have 2 copies at chromosome number 21. But because non-disjunction took place, failure to separate, it took place, now we are left with 3 chromosomes at chromosome number 21, which we call this scenario trisomy 21, which leads now to Down syndrome. So a person with Down syndrome will have an extra copy at chromosome number 21. This you studied only as non-disjunction when you were doing meiosis, but now you learn that it is actually an example also of chromosomal mutation because the number of chromosome has changed. It is no longer what it was supposed to be, but actually it is more number of chromosome at chromosome number 21. Right, so we have come to the end. Just to recap, please remember, you need to know the 3 definitions: mutation, gene mutation, and chromosomal mutation. And then know the examples of each of gene mutation and chromosomal mutations thereof, and how many mutations are beneficial, or the effects of mutation all in all. Harmful, useful, or not beneficial or not harmful, just silent ones. So let's take a look at this example just to apply some of the knowledge that we have learned. This activity, okay, I'm going to read it out to you. It says, sickle cell disease is caused by a recessive allele and first appeared in humans as a result of gene mutation. You must underline that because it gives you a key now. What type of mutation are you dealing with on this question? Now, the table below shows the number of children born with sickle cell disease in some regions in a particular year. Then we must go to the table. We see the region, we have the Democratic Republic of Congo, US, Nigeria, UK, Tanzania, and other, and then you have your total. Now, these here are the number of children born with sickle cell disease. So we have studied our table now. Let's go look at the question now. What is gene mutation? I am actually going to give you 1 minute to come up with a definition for this. Please put your answers on WhatsApp. Alrighty. Gene mutation. Remember, we said now you need to know. The exam might trick you. You must look for the question whether they're asking for gene mutation, they're asking for mutation, or chromosomal mutation. So in this case, it's gene mutation. Then let us look at the definition. Look at where the ticks are. It says gene mutation, it is a change in the sequence. Now the sequence of the nitrogenous bases. So we need to know what is changing. What is changing? It is the sequence of the nitrogenous bases, because all mutations are actually changes, but you need to specify this one is the change in the sequence of nitrogenous bases. That's where you get your full marks. Please don't lose those marks. Check where your ticks are. Then let us take a look at number 2. Which region had the highest number of children born with sickle cell disease in that year? So you're going to come to the table and look at the numbers. So the region that has the highest number, you're going to put it there. So when we look at that, it is Nigeria with 91,011. Right. Number 3, we are now at number 3. With the same table, the same data, it is just a continuation of the questions. Now, what percentage of worldwide total of children born with sickle cell disease came from the Democratic Republic of Congo? Show all calculation. I like calculations. I like this question because it allows you to use the skills in life sciences. Remember, at the beginning of the year in grade 10, you need to learn the skills that you require in life sciences, and calculations is one of them. So in this case, please, I'm going to give you also, again, 2 minutes. I'll say 2 minutes to work out the answer on this one and tell us how you got to the answer. Right. So what percentage of the worldwide total of children born with sickle cell disease came from the Democratic Republic of Congo? So now you want to know, out of the total of the world, how much percentage does the Democratic Republic of Congo consist of, or how much is it? So you're going to take your 39,746, then you're going to divide it, which is this one, this amount here. Then you're going to divide it by the total. And remember, every time when you want to convert something to a percentage, you multiply it by 100. So there's why there's a mark here on 100, and there's also a mark here on the division from what you want from the total. Then all in all, it is 13% of the children in the Republic of Congo that have sickle cell disease. Now, let's come to number 4 quickly. Use the letters capital letter D and small letter d to give the genotype of a person who, A, suffers from sickle cell disease. Now, you need to know, sickle cell disease, is it caused by a recessive gene or by a dominant gene or a recessive gene? So now you must, right. So a person with sickle cell disease, if you remember when you go back to the introduction, to the statement that you got, we were told that sickle cell is caused by a recessive allele. So now that is why a person with sickle cell will have small letter d. So these will be the alleles to represent a person suffering from sickle cell disease. Now, the person who carries the allele but does not suffer from the disease. Now remember, your heterozygote state. Remember, when the small letter d is with when a big letter d is with a small letter d, the big letter d masks the small letter d. Then our person will have an allele for sickle cell, but it will be masked by the dominant one. Therefore, the person will carry the allele, but they will not be affected or they will not suffer. So hence, their alleles are big letter d and small letter d. Now let us take a look at another example. This is a very interesting one. Takes you back to DNA, your protein synthesis. Now the diagram below shows a stage in protein synthesis. Now let's check, okay? Before you do anything, the skill that you need to master is to study the diagram. Even if the question does not say study the diagram, you know how to study the diagram. You must first try to label what is on the diagram, what you see there, and then you can make your own understanding of the diagram. Then you can go to the question. You don't just jump to the question, come back to the diagram. So you make sense of the diagram first. So when you look at this diagram, it shows you a stage in protein synthesis. And then when you try to make the stage here, you see your tRNAs, you see your mRNA, then you must know this is a translation stage. Then, okay, what will be this? This will be my amino acid joined together by a peptide bond there. Okay. Then what is happening? You check all your codons on your mRNA, you check your codons, you check your anticodons on tRNA. You just guess, okay, these are my codons, what will be my anticodons. That is you studying the diagram, making sense of the diagram before you even know what is going to be asked of this. Now it says, the table below shows the tRNA molecules that code for different amino acid. Remember, when you get the table of the amino acid, you must always check in what form are they given. So here it says the tRNA molecules that codes for different amino acid, meaning the name of the amino acid here is going to be given by this tRNA, the triplets on the tRNA molecules, which are your anticodons. Now let's come to the question. It says, in codon 3, you must now come to codon 3, okay? Now you know you are working with this one. Guanine was replaced by cytosine. Now you see UCA. So actually they say initially this C was G. It was supposed to be G, but now it has been replaced by this C. Right, you have now tried to make sense of this now as a result of mutation. So what type of mutation occurred there? You must know. It is gene mutation because it is the change in the sequence of the nitrogenous bases. So you see now these are the nitrogenous bases. You must see the sequence was supposed to be UGA, but now the sequence is UCA. Right. Explain how this mutation will affect the protein that will be formed. So the protein, these are the proteins. So how will this change affect the protein? Most of the learners get this question wrong because they just want to give a simple answer. They just want to go straight to the answer. But you need to unpack it. Show us where it comes from. So let's look at how you must structure your answer. So you say threonine will be replaced by serine, or forming a different protein. How do you know this? How did you get to that? So you must come check. UCA codes for what? You come to the tRNA, but UCA also matches with what first? It matches with A, G, Right. AGU is your serine. Now, serine, here is serine, is going to come here instead of what was supposed to come, threonine. So threonine was supposed to be your ACU. Because of this G, it was supposed to be ACG, your anticodons on your tRNA, because this is what gives you the names of the amino acid that are going to be formed. So now you see, you take it from your codons, your anticodons, what gives you the name, and then or you can say, you can structure your question in this fashion to say UGA codes for threonine and UCA codes for serine, forming a different protein. So the protein that is forming here is going to be different from what was supposed to be formed here. So that is a gene mutation, an example of gene mutation. I don't know if we have time to finish our last question. We can continue with the last question. So this is a question on chromosomal mutation. It is very easy. You have done this with meiosis. So this is a diagram that shows, okay, the diagram below shows a human male karyotype. Very important, human male karyotype. You don't know, you might need that information. So when you study this karyotype, you see the pairs of chromosomes there, you see, and then you see here at number 16, there are 3 chromosomes. Already, that clicks something in your head. And then you move, okay, everything seems to be fine. Now, how many chromosomes does this man have? Meaning you need to count all the chromosomes that this man have. So including the extra chromosome that this man have, okay? Okay, we have the answers for everything now. Now, how many chromosomes does a normal human being have? We know a normal human being is supposed to have 46 chromosomes, but in this case, this male has got 47 because of the extra chromosome at chromosome number 26. Then this man has 3 chromosomes at number 16. This is a result of non-disjunction of chromosomes. What is this type of mutation called? Hence, you need to know the types of mutation. This will be your chromosomal mutation because it happens with the chromosomes with your karyotype. I think that brings us to the end of our session. Okay, we have an extra activity. We can do this one with your teacher. You will see the slides. The teachers will go through this example with you. But please take note of the points that we have given you, the study tips that we have given you, and your exam guideline needs to be by your side every time when you study. Thank you so much.